Animal testing has been the backbone of drug safety research for decades, but now that's starting to change. The FDA now supports the use of new alternative methods, or NAMs, which involve human cells, tissues, and computational models. These tools not only reduce animal testing, but also help researchers understand how drugs behave in the human body before clinical trials. Welcome to Tell Us What You Know, a series by Drug Discovery News. And in this episode, we're joined by Jonny Sexton from the University of Michigan. He's going to tell us how his lab's liver organoids and organ chips are advancing this shift, and how technologies like high-content imaging are powering safer drug development. Let's get into it. Hi, Jonny. How are you doing?
I'm doing great. How are you?
I'm good. We hear a lot about new alternative methods or NAMs these days. What are NAMs and why is everyone talking about it?
Sure. So NAMS is sort of a funny acronym because it doesn't really mean much by itself. But in the drug development community, we all understand that NAMS, new alternative methods, are improved methods to evaluate drug safety. And they include testing in human cell-based systems, organoid-based systems, and microphysiologic systems, as well as computational systems. These days, we still have failures in drug development. The failure rate is extremely high. And one of the main reasons why drugs fail is because they have unanticipated toxicity when we go to first in human studies. And that means that the conventional methods that we're using are not predicting the human outcomes well enough. And so that's why we need new alternative methods to improve that scenario, because drug prices are very expensive. So if we can address that one problem alone, better predicting human drug safety, then we can make drugs much cheaper and much safer for the general population and also for participants in clinical studies.
And I know that your lab is developing some of the new alternative methods, including liver organoids and liver on chips. So, how do you design these models to make them closely mimic the human liver?
Yeah, so the liver is the most important organ for drug metabolism, and liver injury is the number one reported adverse event in terms of drug safety. What we do is, number one, here at the University of Michigan, we have a longstanding clinical study that focuses on characterizing patients who have experienced drug-induced liver injury. So the first step in our process is recruiting and enrolling patients who have experienced drug-induced liver injury. We get a simple purple top, 10 mil tube of blood, and we can extract the white cells from the blood. And from there, we reprogram them into stem cells. They're called induced pluripotent stem cells. Now, once we have these stem cells, we're able to then engineer a piece of that person's liver. We need to look at a diverse collection of stem cell lines that have been established from a variety of patients who have actually experienced the disease. And then the second step is to evaluate a particular drug for toxicity in that engineered piece of liver. So from there, we have a highly qualified, very physiologically relevant model. And depending on the stage of drug discovery, we've developed multiple tools. For early stage drug discovery, we've developed a high throughput 384 well plate assay based on the cell painting approach. We have tested all of the drugs that have previously caused drug-induced liver injury, and we have very high fidelity prediction of drug-induced liver injury from all of the known compounds that have failed due to drug-induced liver injury. And then for later stages in drug development, this is where we need the highest sensitivity in predicting drug-induced liver injury risk. So what we've developed is a liver on a chip system that incorporates many different cell types in the liver and can accurately predict drug-induced liver injury. And what's interesting about drug-induced liver injury is there's not just one mechanism. There are many different potential mechanisms that can lead to drug-induced liver injury. And we're able to accurately predict what different mechanisms may be at play. And that really helps because you want that information so you can go back and either design new compounds that avoid that risk, or it can also inform what to be looking for in terms of adverse events in clinical studies and first in human clinical studies.
Now let's talk about the tools that you use. So how does advanced imaging technology help you study and quantify drug toxicity in these models?
Imaging is really our main tool in our toolbox for assessing drug-induced liver injury risk. As they say, a picture is worth 1,000 words, but our pictures are maybe worth 10 to 100,000 data points. So this approach is called high content imaging. Whether we're using our high throughput method for detecting drug-induced liver injury, or if we're using a microfluidic liver chip, at the end of a drug exposure period, we can then fix the cells and then stain them with a set of fluorescent markers. And after that staining, then we take them to our Yokogawa Cell Voyager 8000 microscope. This is a microscope that's capable of assessing tissue on slides to multi-well plates and for these microphysiologic systems. And at the end of this experiment, we get terabytes of imaging data. So what we do is we observe every single cell that is in the plate or that's on the chip. What that allows us to do is computationally analyze those images to assess the drug-induced liver injury risk. And when we have an image set, we use advanced machine vision software to recognize every single cell that we see and to measure about 6,000 measurements for every single cell. Now, when we screen a set of those compounds, those drugs that we know already cause drug-induced liver injury, they perturb the cells in a particular way. So if it's a hepatocellular injury drug, they may cause mitochondrial toxicity. Well, MitoTracker is one of the dyes in our set, and we can directly observe if there's any impingement of mitochondrial metabolism, and that is a definite warning signal. So we need to see that on a per cell basis so that we can see, is this affecting hepatocytes, cholangiocytes, or hepatic stellate cells, or other kinds of cells that are in our system. So once we have results from a new compound, then through what we call morphologic profiling, we can compare all of those 6,000 measurements and see if we can get a match to a known DILI-causing drug that has a known mechanism of action. And that is one of the ways that we ascribe the risk.
And now can you share an example where one of your models successfully predicted a toxic effect that traditional testing may have missed?
Oh, definitely. Yeah. So we published a paper just last year on a drug called inirigavir. In this particular story, the phase 2A study that administered inirigavir at about 200 milligrams daily showed no real substantial risk. There was some moderate elevations of liver enzymes, but it wasn't anything that the hepatologist in charge was going to halt the study for. And so what happened then is that drug progressed to a phase 2B clinical study where they did dose escalation. So they went up to 400 milligrams, so they doubled the dose, and the drug, this is for chronic hepatitis B, was administered with a standard of care drug called tenofovir. As it turns out, the combination of these drugs was synergistically toxic. Either one of them on their own appeared safe, but when you put them together at the right doses, it unfortunately caused serious drug-induced liver injury in all the patients before they halted the study. What we did was, because this was such an important study, we used our liver on a chip system and looked at tenofovir alone and inirigavir alone, and we observed no toxicity. When we put them together, and this was at the clinically relevant concentrations, we saw a very prominent synergy in the toxicity mechanism. And through characterization using imaging with the Yokogawa Cell Voyager 8000, we were able to detect the risk. We determined that the risk was very prominent, and also it was within the clinical dosing regimen. We observed that there was mitochondrial toxicity, as I was mentioning before, but only in that combination. So this is also a really important technology to detect drug-drug interactions. And because of our microscopy system, we have, I call it the world's best 20x objective. And it is very high resolution, and it has a very long working distance. And so that's really important for chips because we have one cell type on the bottom and we have different cell types on the top. So it's a multi-layered chamber. And through optical Z-sectioning, we can image cells in this layer in the bottom compartment and also cells in the top compartment. So the advanced microscopy really helps us to interrogate these chips. The other thing is that the dual microlens spinning disk confocal system allows us to have high clarity in the bottom compartment and also in the top compartment. So we get high resolution, beautiful images in both compartments because of this microlens confocal unit. And the other amazing thing about that unit is that it allows us to do live cell studies as well. We can do this imaging while the cells are alive on the chip because this confocal system dramatically reduces the phototoxicity. So that's one of the things that we're working on now is to try and do time series kinetic studies so we can understand after exposure, when does the risk really present. And it's wonderful to have a system that's capable of doing high resolution imaging on even live cells where we can deliver the temperature and humidity environment as well as 5% carbon dioxide, which is what they need. And so it's like a full featured microscope inside of an incubator.
So lastly, tell me what's next for your research and what do you think needs to happen for NAMs to be more widely adopted across the pharmaceutical industry?
We've been talking a lot about the experimental systems, but we're also equally focused on building artificial intelligence models that can then ultimately predict the drug-induced liver injury risk directly from the chemical structure. So we have screened thousands of compounds now through our high-throughput screening system, and we understand that some of them cause drug-induced liver injury clinically, and some of them don't. So we have positive and negative controls. And through this, we're able to take all of those 6,000 measurements for millions and millions of cells in these images that we obtain. And we can train artificial intelligence models to enhance our predictive accuracy. Then in the future, we're very excited to combine different organ systems like as in connecting chips from different organ systems. We think it would be very cool to connect a gut chip with a liver chip so that we can actually administer the drug to the gut side, watch the transport, watch the penetration into the liver compartment, and then evaluate the toxicity potential there. Because now that can give us even more information about exposure plus toxicity. That's where we can really advance these models to the level of robustness that they need in order to be adopted by the drug development community and also, most importantly, the FDA. So the second part of your question is, what is it going to take to implement these novel alternative methods in preclinical, non-human laboratory testing that can actually be used and submitted to the FDA in an IND data package? You have to do a prescribed set of tests, and one of which is determining liver toxicity. Each lab has a slightly different method for producing them. So we need standardized methods. They need to be qualified and validated, across multiple different sites. and this is so that they can be run in a GLP good laboratory practices environment. And that's what it takes in order for the FDA to rely on this data, as to support the fact that this drug is not going to injure humans when it goes into clinical studies.
Indeed, that's such an exciting and important direction in the future. Well, thanks again for your time and your insights with us.
Thank you.
If you enjoyed this episode, remember to like, subscribe, and check out our other videos for more expert insights. I'll see you next time.